So I'm looking for Robbie. Not here yet. Luke, also not here yet. I still do not see the first speaker. That is worrying. That worries me. Is Luke in the Zoom room? Luke, are you here? I don't see Luke. And I don't see Robbie. We're still waiting for the first speaker to appear, but the last 3 speakers are already here. Chart, you know Luke, right? He's from UP.
Hi, Lawrence. Yes, Luke is my student. He's from UP. That's right.
Okay, I don't see him here.
Well, his username is Walter.
Oh, Walter.
Yeah, Walter Chinedu.
Oh, okay, he's here.
Yes, that's him. Yes.
Right. Okay.
If the first speaker is not there, then I guess Luke could start the session.
Yeah. So Luke, I've made you a co-host. Why don't you test your share screen, and we'll give the first speaker another 2 minutes to show up. Thank you. Looks very nice. Yes, we can see that. Okay. So let's see. Let's give it another 2 minutes for the first speaker to show up. Even if he showed up now, he'd be 5 minutes late. Let's see. Maybe the registration system should ask for people's cell phones so that we could either call or send a text. I remember I had a professor who was, I don't know, we could have class at 3 o'clock and you could be talking to him at 10 minutes to, and he would still forget that he had class at 3. So it always took someone to go to his office and get him.
Yeah, and if I can make a quick comment as well. So sometimes on the registration, on the conference website, so the times are sometimes displayed in local time and sometimes in GMT +2 times. So I don't know if this is just me or if that happened to a few other people.
Oh yeah, it happens a lot. People, that's why we try to universally use GMT +2 in all the communications. But
yeah, I mean that is yeah, easier for everyone to just convert to their local time. But yeah, on the website. So if I go to timetable, so it's showing me in my local time. But anyway.
Right. Yeah, and that is not as useful as a feature, I think, as that the programmers think it is.
Yeah. Yeah. True.
Yeah. Okay. Well, I guess I would not be surprised if Robbie is maybe has some difficulty in the time zone translation. So let's start with Luke, who's going to talk to us about modeling of Plasmon-enhanced fluorescence. Luke, are you ready? And the talks are being recorded and live streamed, and so keep that in mind. Okay, Luke, go ahead, please. Can you hear me? I think you need to unmute yourself because we cannot hear you. Yeah, you're muted. So,
okay, yeah.
Okay, here you are. Yeah.
Okay. Good day, everyone. So my name is Luke Ugoke. I'm a student at the University of Pretoria, Department of Physics, and I'll be talking to you today about plasmon-enhanced fluorescence and how it occurs in a hybrid system made up of a light harvesting complex and gold nanorod. All right, so the phenomenon involves two stages. So in the first stage, what happens is that if you have a fluorophore or a fluorescent molecule close to a nanostructure as depicted in this place, in this figure, you see the dipole. If the dipole of the molecule aligns in a particular direction, with respect to the surface of the nanostructure, it can lead to an enhanced excitation rate. Okay, and this is due to modulation of the optical density of states in the molecule, and this is primarily caused by the scattered field, the scattered field around the nanostructure, the surface of the nanostructure. And it's a local field in the sense that it kind of decays with distance. It basically goes to zero at long distances. So it's very localized around the nanoparticle surface. Okay, and the second stage, which I'll see go in detail into the stages. In the second stage, you have what we call quantum yield enhancement. And what happens here is that usually the intrinsic quantum yield of a fluorescent molecule is enhanced, okay, when it is close to these nanostructures like nanorods or nanospheres. And one of the, why this is possible is because the noble metals that are usually used are not, they don't chemically interact with the molecules. Okay, but they can interact optically with these molecules and change their optical properties. Okay, so it makes it very interesting. So what you have here, for instance, is a crystal violet molecule, and its quantum yield can actually be enhanced from as low as 2% to as high as close to 1.7% here. And this is just by coupling it, bringing it closer to the tip of a gold porous spheroid. Usually the overall enhancement process is a product of the two stages. The first stage where you have excitation rate enhancement, and then the second stage where you have quantum yield enhancement. Now, this has a lot of applications, and for instance, the excitation stage is very much used for in surface enhanced Raman spectroscopy, where it is used to analyze different kinds of molecules and the optical properties, or analyze that bound to metal surfaces. And this is because the SAS intensity is proportional to the magnitude of the scattered electric field around the surface of the nanostructure. And the application is in plasmonic light emitting diodes, where especially in organic light emitting diodes that use fluorescent molecules as the major absorber of light. So those chromophores, if their quantum yield is weak, then it can be incorporated alongside with metal nanoparticles to enhance their quantum yield. And then also in solar cells, there are lots of research now on plasmonic solar cells, because these solar cells have a very high absorption cross-section. And this can lead to enhancement of the photocurrent density of the solar cell. So the first experiments and simulation studies on this kind of hybrid system was performed using light harvesting complex in 2004. So this was done using light harvesting complex 2 of purple bacteria. So prior before this work, there have been different reports on plasmon-enhanced fluorescence involving dye molecules like crystal violet, fluorescein, and all that. But these researchers were able to actually show that this can also be achieved even when you have light harvesting complexes. So this LH2 is a light harvesting complex of purple bacteria, and it has an intrinsic quantum yield of around 0.1. So it has a very low intrinsic quantum yield. But on coupling this to a gold nanorod, they actually found out that the quantum yield can be enhanced, as you can see here, from as low as 0.1 to around 5 times its original value. And what happens is that usually a particular nanorod length that the extinction cross-section overlaps completely or partially with both the absorption cross-section and the emission cross-section of the fluorophore in question is used. So here, for instance, a nanorod length of 160 nanometers was used in this experiment, and usually in the excitation stage, this citation rate is calculated at this wavelength. For this fluorophore, it was at 800 nanometers, and then at the emission stage, the quantum yield enhancement is calculated. So here it was at 870 nanometers. And then the overall fluorescence rate enhancement, emission rate enhancement, is a product of these two. And that's what you see here. And you can see that it's actually, because this local electric field is what determines the citation rate enhancement. And because of that, it also affects the overall emission rate enhancement, such that at a particular distance, it saturates and it starts to decrease if you move away if you keep moving away from the nanostructure, then start to get like very low values, as you see here. So there's an optimum distance for these enhancements. And as you can see here, simulations were performed as well as experiments, and they were really good agreements. Okay, now in our group, a PhD student that graduated actually did something really interesting, where in this case he studied light harvesting complex 2 of plants, which is the major light harvesting complex in plants. It has an intrinsic quantum yield of around 26%. And like you see here, the nanorod size that was used here is around 80 to 90 nanometers in length. And there is a very good agreement also when you look at the spectral overlap. So when you look at the extinction cross-section or the absorption cross-section of the nanorod and that's of the light harvesting complex, you see that around this shaded region, there is a very good overlap. And this resonance here for the nanorod is due to the longitudinal resonance. So it's along the long axis of the nanorod. So this is to ensure that there is strong coupling between the molecular excitons in the light harvesting complex or in the fluorophore and the localized surface plasmons of the nanostructure. And what you see in this diagram is the immobilized nanorods on a glass substrate. And on this insert, you see that the coupling phenomenon actually leads to, for instance, you see more photon counts for molecules near the hotspots of the gold nanorods compared to molecules that are not close to the hotspots. And this I will explain later is due to something called the Poisson effect. So you see this Raster scan here also showing that complexes near the hotspots of the nanorods are more bright. Okay, they're brighter compared to complexes that are off the hotspots. And hotspots are basically regions where the concentration of the surface charges around the nanostructure is such that it leads to this so-called lightning rod effect, where the scattered field intensity is very high. So these regions are usually called hotspots. And what you see here in this decay plot here is basically the decay rates of the molecule in the presence of the fluorophore or the complexes, in the presence of the nanorod is reduced. Okay, so the decay lifetimes, I mean, is reduced. Okay, and this reduction in the decay lifetimes is due to an increase in the radiative decay rates of the fluorophore. And this phenomenon is what is known as Poisson effect, which I'll talk about later on. So you can see the parameters reported here, which are the enhancement factors obtained at this distance. So another motivation to do this work, because this is a theoretical study that I did, and another motivation was to look at it from a different perspective, which involves some ideas in classical plastonics, where here the fluorophore is not just treated as a point dipole. So usually in the calculations that we do to determine these enhancement factors, the fluorescent molecule or the fluorophore is usually treated as a point electric dipole source. And then we use this semi-classical theory of radiation to determine the decay rates of the molecule of the fluorophore in the presence of this nanostructure. So here you have a homodimer, and then you have this nanocrystal. This is cadmium selenide nanocrystal sandwiched at the gap of this homodimer, and then is studied using two different classical theories. So like in the first one, which I labeled the molecular point dipole model, MDP, the fluorophore is treated as a point electric dipole source, which oscillates and emits radiation. And then in the dielectric nanoparticle model, it is treated as a dielectric nanoparticle that has a particular polarizability. And what that does is that it actually accounts for the exact shape and size of the fluorophore. Usually in the point dipole model, the shape and size of the model of the fluorophore is not taken into account. Okay, so these authors were able to show that there is a huge difference between these two approaches, right? So if you the plot shown here shows that when they interact optically, what happens is that at some wavelengths or energies, you can actually have a splitting in the hybrid spectrum. And this splitting, usually called Rabi splitting, is actually an evidence of strong coupling. So strongly coupled optical systems usually display this kind of splitting in their spectra. So what happens then approach to study or to determine enhancement factors? So if you look at this plot here, it shows that at this region where you have a dip, where you have the splitting, there's a big difference between the excitation rates enhancements predicted by the molecular point dipole model compared to the one that is predicted by the dielectric nanoparticle model. And this is something I wanted to also account for in my model. So I wanted to look at how do I make sure that I can account for this and really see whether it has effects on when you have light harvesting complexes this time around, and not just this kind of nanocrystal. So the major finding here was that molecular shape and size does affect plasmon-enhanced fluorescence parameters. So in my own model, I approximated the light harvesting complex 2 as a spherical dielectric nanoparticle with a certain equivalence for volume radius, and it's at a certain distance from the tip of the nanorod, and the electric field source. In the case of the point dipole model, the molecule or the fluorophore is the electric point dipole source. However, in the dielectric nanoparticle model, the nanoparticle is treated as interacting using via a dipole interaction with the fluorophore in this case. And these are some of the parameters we use here. These parameters actually correspond to the one used in the lab as well for the length of the nanorod and the width as well. So then we used a model known as modified Jesteninsm model. So this model was proposed in the 80s to determine the decay rates of molecules interacting with spheroidal nanoparticles. And usually we add corrections, size-induced corrections, because beyond a certain size, then such models are not valid unless you modify it. So that's why it's called the modified Jesteninsm model. So which I'll talk about. So these are most the equations, the key equations that we're driving this work. So the first equation here is basically the molecular polarizability of LHC2, and this was modeled using critical point functions, which basically looks at the different molecular resonances in the nanostructure, in the molecule, and then is assigned some phase functions and also some exciton line widths are also assigned to this function to account for these line widths that are in the absorption spectrum of the complex. Usually this kind of molecular polarizabilities can actually account for the ground state absorption of the molecule in question. So in equation 2, we have the excitation rate enhancement, which, like I mentioned before, depends on the local field. And this local electric field also depends on the scattered field from the nanostructure. Okay, so it's calculated as follows, and then the radiative rate enhancement, known as the Poisson factor, depends on the induced dipole moment, which is calculated as shown. And then you have effective polarizabilities due to this interaction between them. And then you have this non-radiative decay term, because the molecule undergoes both radiative and non-radiative decay. So it's calculated using these expressions. So our major result looks as follows. So in the excitation stage, we can see that when the molecules are not, when the system is not coupled, so you have the uncoupled system, you have the nanorod is on its own, is not interacting with the light harvesting complex, then you can easily see now that there is a very good overlap between the peak of the plasmon spectrum and the peaks of the molecular spectrum, as you can see here. And also our analytical model of the molecular polarizability agrees well with the experimental data. Now, when they couple together, they form a hybrid system. And this hybrid system, we have its own spectra. Okay, and you can see here that this spectra is very sensitive to the distance, the surface-to-surface distance between the molecule and the nanorod. So at very short distances, the interaction is so strong that it induces these dips in the spectra. So these dips are evidence of strong coupling. And now as you move away from the surface, then the interaction weakens and it enters into the weak coupling regime where these dips begins to vanish. So now we can actually calculate the excitation rates via these two different models. So in the Plasmon approach, the dielectric nanoparticle model, you can see that it's is less than the molecular point dipole approach, because what you notice now is that we are taking into account the shape and size of the fluorophore. We took that into account. So because of that, you have something that is actually less. But this is only true if you are close to the surface of the nanorod. Away from the surface of the nanorod, those two models are actually in very good agreement. And compared to experimental data, we can see that this is more than twice what was predicted in the lab. And we attributed this to the presence of dielectric coatings around the nanorod surface that was used in the lab version, which we haven't yet accounted for in this theoretical model. And then the radiative decay rate enhancement, also known as the Posey factor, is calculated at the emission, peak emission wavelengths of this light surfacing complex. And as well, you can see that there is a big difference between these two models, especially at short distances. Also compared to experimental data, you can see that this, the Posey factor P, that was calculated theoretically is.
Luke, can you take five more minutes?
Okay, 5 more minutes. Okay, so, and then we went further to calculate quantum yield enhancement as well as emission rate enhancement. So you can see the line here shows you the quantum yield enhancement. I mean, the intrinsic quantum yield of the fluorophore, which was 26%, and now at certain distances, you can notice enhancement, and then you move away, and then it starts to approach the intrinsic value once again. And then the overall emission rate enhancement is shown as follows. The two models also predict different factors at short distances, but at distances greater than the size, the molecular size of the complex, that these two models are the same. So the conclusions from this was that this interaction induces hybrid resonances, which are called plastonic resonances in the spectra, especially at short distances, which is an evidence of strong coupling. And also the two models actually only agree at long distances. At short distances comparable to the molecular size, then it's actually more interesting or more accountable to take this shape and size of the molecule into consideration. And then the enhancement factors from the two models are less than experimental value. But we attributed this to mostly the fact that we are yet to account for dielectric effects and also some retardation effects induced by size-dependent damping and dynamic polarization of the induced surface charges around the nanostructure. We are yet to account for this term, for these effects in the non-radiative term. And then hopefully after this two, we can then perform simulations also to see which one works better when compared to experiments. So I thank my supervisor, Prof. Judge Krug, and my co-supervisor, Prof. Thomas Mancha, and the entire Biophysics and Photon Research Group at the University of Pretoria, and my funding sources are University of Pretoria and the National Research Foundation. Thank you for listening.
Thank you. Are there any questions for this paper, this talk? Let me see any hand raisings. We will do a hand clap. So I have one question, I guess. So the gold nanorod, would you consider that an enhancer or an inhibitor?
Yeah, so it can be an enhancer or an inhibitor, depending on how close you are to the nanoparticle surface. And this is why in the lab, they usually coat the surface with some dielectric, because if the fluorophore interacts directly with the metal surface, it can induce, lead to surface-induced quenching of fluorescence. Yeah, so that distance is always a problem in the lab. Experimentalists always battle to control that distance to find an optimal distance for the enhancements to take place.
Okay. Any other questions? So I think the first speaker might be here by now. Maybe not. I got an email from him. So we'll go next with Domsey. Are you here? Domsey was here earlier. Yes, I see you. So what we'll do is, if you could stop sharing your screen, Luke, and then we'll go with the next speaker, Domsey.
Thank you.
Let's applause again the first speaker. Yes, we can see your slides now.
May I begin?
Yes, please.
Good day, everyone. My name is Namso Kumisa. I'll be presenting my work in silico study with the title Epileptin-Derived Identification of Potential Inhibitors of Schistosoma mansoni HSP60 towards Anti-Schistosomal Drug Design. Neglected tropical diseases are a group of debilitating diseases that are associated with the poor, and they're highly common in third world regions of the country. Schistosomiasis is the second most prevalent neglected tropical disease and is caused mainly by 3 species: Schistosoma japonicum, Schistosoma hematobum, and Schistosoma mansoni. Transmission of the disease is highly prevalent in areas with water activities such as agriculture and fishing, as well as in women and children that perform everyday duties in contaminated water sources. The global distribution of schistosomiasis can be seen in the world map above. As you can see, Schistosoma mansoni, Schistosoma hematobum have the highest prevalence in the sub-Saharan African region. And closer to home, Magaiza et al. Has indicated that over 4 million South Africans suffer from the disease. And this is particularly true for areas that are involved in agricultural activities, such as Limpopo, KwaZulu-Natal, Mpumalanga, and the Eastern Cape. The life cycle of a schistosome is divided into 2 hosts and begins when the eggs of the worm are excreted in water sources by an infected host. The eggs in the water develop to myricia that can infect different snail species. And within the snails, they undergo asexual reproduction to become pathogenic cercaria that are excreted back into the water, where they can infect a human through penetration of the skin, becoming schistosomia within the human host and traveling through various organs, such as the liver, heart, and lungs via the veins of the human. In the hepatic portal vessels, the male and female worms pair up and travel to various regions, such as the bowel region and the bladder, to where reproduction occurs. And the eggs that are produced can be retained in these tissues, resulting in disease manifestation. In the gastrointestinal region, resulting in gastrointestinal schistosomiasis or urogenital schistosomiasis. The symptoms that can be seen in patients with schistosomiasis include Katayama fever, diarrhea, and as well as in the case of urogenital schistosomiasis, hematuria can also be noticed. All species of Schistosoma mansoni are associated with granuloma formation, which is associated with the development of colorectal cancer as well as bladder cancer. Schistosoma hematobum, which is the causative agent of urogenital schistosomiasis, is associated with also associated with an increased risk of HIV as a result of the sandy patches that form in genital regions of females, resulting in an increased physical barrier during intercourse. There have been various treatment options against the disease. Antimony salts, oxamnicrine, and artemisinin and its derivatives have been used. Unfortunately, antimony salts and oxamnicrine have since been outdated due to their associated side effects, while artemisinin and its derivatives are mainly focused on antimalarial drug design. The main or cornerstone treatment for schistosomiasis has since been praziquantel. The drug has been used for over 30 years and has been effective due to its ability to work on all schistosome worms, or species rather, as well as its mild effect on patients. Despite being used, or despite the prolonged clinical use of the drug, the mechanism of action of the drug is still unknown in the scientific community and has led to the most accepted hypothesis is that ingestion of the drug by an infected individual results in the bioavailability of the drug to the worm. Ingestion of the drug by the worm results in influx of inorganic ions, particularly calcium through the voltage-gated calcium channels, resulting in muscular paralysis, as well as vacuolation of the tegmental structures of the worm, which results in antigen exposure and therefore clearance by the host immune system. Unfortunately, the prolonged use of the drug, which has been used for over 30 years, has made it susceptible to resistance. The resistance can come about through the ABC transporter proteins, is associated with ABC transporter proteins, which will result in an efflux of the drug once it is ingested or made available to the worm, resulting in no influx of inorganic compound of inorganic ions, and therefore survival of the worm within the host. This has necessitated the development of new anti-schistosomal drugs, as the various biological products that are produced during the worm's life cycle provide an array of biological targets that can be used against the worm. A notable, or one of the most significant changes that occur is its transition from a cold-blooded worm to a warm-blooded human host, which results in an increase in temperature, causing thermal stress within the worm and resulting in the degradation of various proteins that are important to the worm. To fight this, the worm itself has an inbuilt response that is common, called the heat shock response, which results in an overexpression of a group of highly conserved proteins called heat shock proteins that are involved in the refolding of major proteins within the mitigation of the proteotoxic environment caused by the thermal stress. Heat shock protein 60 is among various proteins that are produced within the worm. It has been shown to be expressed constitutively and during stress in the worm, and is a vital component in folding of the damaged proteins that occur during the thermal stress. It has also been shown by Masamba et al. To be a possible interactor of universal stress proteins that are also produced in the worm during this difficult time. The protein itself works by taking in an unfolded protein, and in the presence of an ATP molecule and a co-chaperonin HSP10, they create an unfolded cage wherein protein folding occurs. And once the protein has been folded, it is released, and an ATP molecule and HSP10 and the HSP10 are also released to provide an HSP60 that can take up more unfolded proteins to refold. The significance of this protein in this research study in particular, the schistosoma mansoni HSP60 protein was used to determine it was used as a biological target to study or to find anti-schistosomal drugs. The aim of the study itself was to derive potential inhibitors of schistosomiasis HSP60 from a known inhibitor that was identified by Nagumo et al. In 2005 called epolilactine, and to computationally characterize a putative interaction between the protein itself and those derived inhibitors. The aim of the project or the research was achieved using the above objectives. The study itself was mainly in silico, and it began with modeling of a 3D structure of schistosomiasis HSP60. This was followed by taking a structure of epolilactine and using the relevant pharmacophoric moieties into zinc pharma to derive structural analogs of the drug or compound. The obtained compounds or hits that were obtained through the zinc pharma server were modeled or docked to the 3D structure using autodock. Those that had the lowest binding energy, that showed the lowest binding energy during the docking, were used for subsequent molecular dynamic simulations, and the trajectories that were obtained in the simulations were thereafter analyzed. The first part of the results shows a 3D homology model of SMHSP60 that was generated from a 73% sequence identity to the human mitochondrial HSP60. Figure B shows the Ramachandran plot that was used to verify that the 3D model that was generated did not violate any stereochemical parameters. Figure A shows the pharmacophore model of epolilactine that was used during the search for other inhibitors, and figure 1, 2, and 3 show the potential inhibitors that were derived as well from the zinc pharma as well as their zinc IDs. The complexes shown in ABC show the ligands that were derived from epolilactine within the binding cavities of this protein itself, as shown in the close-ups, as well as the docking complexes. The 3 compounds, which were among 7 that had been obtained from zinc pharma, had the lowest binding affinities, as shown in the docking scores above, and these were the ones that were subsequently used for the simulations. The simulations that were run, were analyzed, the trajectories were analyzed, and they began with determining the root mean square deviation, which is an indication from the original SMHSP60 structure, which is shown in black as the upper structure. The simulations that were run, all the simulations were run for 200 nanoseconds, and all the complexes as shown with the derived inhibitors were compared to that of epolilactine, the original inhibitor. All the complexes were shown to reach stability after 100 nanoseconds, and the table alongside the graph is an indication of the average RMSD values that were obtained for each of the complexes in comparison to that of the EPO structure of SMHSP60. The residual fluctuation were also shown in the RMSF graph. The fluctuations, the highest, the compounds with the highest, or complexes with the highest residual fluctuation were shown to be epolilactine, bound to SMHSP60, as well as inhibitor 3. Inhibitor 2 was shown to have the lowest residual fluctuation, and certain regions of relatively low dynamic fluctuations that were shown in the graph, as shown in the graph, could be an indication of the residues that are important during ligand binding, as well as the structure stabilization during binding. The compactness of the structure was determined using the radius of gyration. As shown in the graph, all the structures reach compactability, but system 2 had the highest average ROG, while system 3 and system 2 in the epolilactine bound structures had system 1 and system 3 had the closest structure to the upper structure, compactness of the upper structure, as shown in the table alongside the graph. The hydrogen bonds that were formed during the simulation are shown in the graph. As shown in red, the epolilactine has showed the lowest formation of hydrogen bonds during the simulation, while system 3 or inhibitor 3 had the highest number of hydrogen bonds formed, which was also consistent throughout the 200 nanosecond simulations. Table 1 is also an indication of the total binding free energy, which allows us to identify the strength of activity as well as structural interactions of the potential inhibitors and their potential as pharmacological targets. As shown in the table, and particularly in the one highlighted in the structure highlighted in red, the total binding free energies were indicated that inhibitor 2 and 3 showed the lowest binding free energy, while apolilactin and inhibitor 1 had the closest, inhibitor 1 resembled the binding affinity of apolilactin of the original compound. The energy profiles the following energy profiles highlighted the energy contributions that were relevant to the total binding free energy and the interacting amino acids of SMHSP60 once the compounds bound to the structure. The graphs themselves indicate a conservation of aspartic residues throughout all 4 complexes, which could indicate that the binding pocket itself had an overall negative charge, and is an indication that the residues that play the highest energy contribution, and their participation in the formation of important bonds during ligand binding. In conclusion, from the compounds that were derived from apolilactin, and they are in comparison to apolilactin itself, the derived inhibitors showed a higher enhanced activity against the SMHSP60 protein. System 2 and System 3, or inhibitor 2 and 3, showed the highest activity and had the highest potential as lead compounds in anti-schistosomal drug design, while the key interacting residues that were shown in the energy profiles and that contributed to the low binding energies of both System 2 and 3 could be used in the design for the search of other potential inhibitors of SMHSP60. These are my references, and thank you.
Thank you. That's a very, very nice talk. Are there any questions?
Yeah, I have a question.
Yes, thank you.
Yeah, so impressive talk, of course. So I was wondering if you had run a simulation of the same compounds against the human HSP60. So the reason why I am asking is because obviously you want to have something that is specific to your target.
So the simulations were not run for the template that was used to generate. The human mitochondrial HSP60 was just used as a template using modeler to model the 3D structure of the schistosoma mansoni HSP60 itself, because it does not have a defined structure. So it was that was why we used that. But no, we did not run simulations on the human mitochondrial HSP60 itself.
I have a quick question. So did you run a calculation on the electrostatic nature of the binding pocket using, I guess, any number of like molecular electrostatic packages?
Pardon? Can you please repeat that? I couldn't hear you properly.
Were you able to run, or have you contemplated running any molecular electrostatic calculations on the system to see if the binding pocket does actually have a negative electrostatic potential?
No, it has not been run. The only thing that we're trying to determine actually from the study was whether or not the compounds that we would derive, their activity as compared to the original inhibitor of HSP60. So to check, that would require us to do ANS, which would be an experimental evaluation, which could be done experimentally when we do experimental part of the project itself, to determine like regions of the binding pockets within the whole protein itself. So no, they have not, simulations of that nature have not been run, but it could be done experimentally through ANS to determine binding pockets within the protein itself once it has been expressed.
Okay, thank you. Are there any other questions for this speaker? All right, let's give her some applause. I keep using this applause button in hopes that I will eventually find the raise hand button, which I don't know where that is still. Before we move on, I got an email from the first speaker, Robbie, but have you come to the Zoom room yet? Still not here. That is odd. Okay, well, let's move on to the next speaker, which is Delay.
Yeah.
Did I let me make sure that you are able to. Right, you're able to share your screen now. Thank you.
Yeah. Sorry, just
Fantastic. Looks beautiful. Thank you.
sorry, just sorting this out quickly.
Okay, thank you. Go ahead.
Perfect. All right, so okay, so good afternoon. I will be presenting my research on with the title Molecular Simulations of the Interaction between Schistosoma mannsoni, actinyl-dynein intermediate chain protein, and dynarestin. Okay, this is just a brief overview of the topics I'll be covering throughout the presentation. So praziquantel was discovered in the 1970s and has been used to treat trematode and cestode diseases, including schistosomiasis, for over 30 years. And the drawback of using this PZQ is its inability to prevent reinfection, as well as its inability to kill the juvenile stage schistosome parasites. The mass administration of this drug subsequently led to the evolution of drug-resistant parasites, thus creating the need to find new inhibitors against the identified actinyl-dynein intermediate protein target. Schistosomiasis is most prevalent in developing countries, mainly tropical and subtropical countries. Now, figure 1 indicates the widespread prevalence of all of the schistosome disease-causing parasites. However, emphasis on S. mannsoni will be placed, and as can be seen, is most often found within Africa, South America, as well as the Middle East, as well as the West Indies. The annual mortality rate is currently around 300,000 individuals, and the global infection rate has been estimated to be 206 million, with approximately 90% of that population residing within Africa. But of that 90%, approximately 5 million individuals in South Africa, within the KwaZulu-Natal, Mpumalanga, and Limpopo areas, and spots in Gauteng and Cape Town as well, are infected with this disease. So schistosomiasis is more commonly referred to as bilharzia and is caused by 3 main schistosome parasites, being S. japonicum, S. hematobium, and S. mannsoni. Now, the S. mannsoni eggs are easily distinguishable from the eggs of the other species due to its protruding lateral spine over here. Following the initial infection with the parasite, infected individuals develop Katayama syndrome as an early manifestation of schistosomiasis, of which the symptoms may include fever, fatigue, and malaise. Now, S. mannsoni has 2 stages in its life cycle. The asexual stage begins within a freshwater environment in which the eggs have already been released. Now, these eggs hatch to form miracidia, which swim and infect their respective snail host of genus Biomphaleria. Subsequent to this, these miracidia develop into sporocysts, which then reproduce asexually to form Sacariae, which have forked tails and are released back into the freshwater environment. Now, the sexual stage begins upon the penetration of the mammalian host skin by the Sacariae, which then shed their tails and become schistosomulae. They then enter the host circulation and migrate to the portal blood in the liver, where they mature into adults, and the male and female worms pair off together and then migrate to the small intestine where the female lays its egg to ensure the continued parasitic life cycle. According to Aruleva and colleagues in 2019, the first drug treatments discovered included metrophonate, alteplase, and nirotazole. However, these were discontinued as they required numerous and frequent doses, caused intense nausea and pain, and were shown to be carcinogenic respectively. Alternatives to these, such as oxamniquin, mefloquine, and artemisinin, were then introduced as slightly more effective treatments. However, each of these had their own setbacks being used. So since the decline in effectiveness of the PZQ drug, newer, more cost-effective treatments have been sought after. The axanemaldyene intermediate protein thus presents itself as a new target for inhibition. Now, this protein consists of multi-subunits, which consists of heavy, intermediate, and light chains, and they all play a role in cilia and flagella beating, which is to ensure the motility of the actual parasite. So inhibition of this target protein by a novel dynein inhibitor known as dinerestin may lead to significantly reduced infections. Dinerestin was synthesized in 2018 by Suzanne Hoying and her colleagues, whom tested it against cytoplasmic dynein. Now, through chemical alteration, by the addition or elimination of functional groups at various points on the molecule, this drug may be used to inhibit the axanemaldyene protein target. Chemically altering the molecule would increase the binding potential between dinerestin and the protein target. And figure 3 here just indicates the chemical structure of dinerestin, represented by figure 3A, and then 2 of its analogs, which are B and C, being less active and inactive respectively. So the aim of this research was to identify the binding affinity, homology, and molecular dynamic characteristics of the starting protein inhibitor dinerestin on the S-Mansoni axanemaldyene intermediate protein using various bioinformatics simulations. My hypothesis was that dinerestin would portray novel inhibitory characteristics and a strong binding affinity for either the active site or an allosteric site on the axanemaldyene intermediate protein. Okay, so the protein was modeled using the built-in modular functionality of the Chimera software, and the proposed model was then uploaded to the ProCheck server where it was processed and a Ramachandran plot was produced. Hydrogens were then added to the inhibitor and deleted from the protein to prepare the ligand and docking for docking onto the protein. Molecular docking was then performed on the docked complex by utilizing AutoDock-Vina to identify the approximate binding sites for ligands to the protein. Following successful docking, the protein and ligand were allowed to interact for 60 nanoseconds, and post-MD analyses were then performed on the results. So for continued studies, pharmacophore was generated by uploading the dinerestin structure, as well as the generated pharmacophore to the Zinc Pharma server to screen for hits, which could be employed for in vitro testing on various schistosomal cell lines in future interaction studies. Okay, so the modeled protein consists of 11 beta sheets and 1 alpha helix, as seen in figure 4A. The Ramachandran plot provided an assessment of any violated stereochemical parameters of the modeled protein and indicated that 84.7% of the homology model fell within the most favored region, which are these red regions over here. The 17 residues making up 13.7% of the model, which fall into the additionally and generously allowed regions, which are just these yellow and light yellow regions respectively, add to the main value with a total of 98.4%. Now, this represents a good homology model as it is above the 95% suggested homology to continue studies with a modeled protein. The 2 residues which lie in the disallowed region only make up 1.6 of the homology, these 2 residues over here, and are thus negligible. And the clustering of dots or residues within this red regions over here indicate the formation of secondary structures within the protein. So they could either be alpha helices or beta sheets. Following this, the binding affinity between the inhibitor and protein was assessed, where figure 5 indicates the residues the inhibitor might interact with upon binding to the active site of the protein. Now, 2 of these interactions are with hydrophobic phenylalanine 122 and leucine 111. residues, which are indicative of the inhibitor binding to an active site situated within the core of the protein. Other interactions include two polar interactions with serine 115 and serine 123 residues. Now, a Z score or a binding score of -6.2, as seen in table 1, was also obtained, which indicates a very strong binding affinity of the inhibitor for the modeled protein. Subsequent to molecular docking and allowing the protein and inhibitor to interact for 60 nanoseconds, post-MD analyses of these results were required. The RMSD indicated the measure of stability both prior to and after the inhibitor had bound to the protein. This averaged value changed from 3.12 angstroms to 3.69 angstroms respectively. This means that the protein had been destabilized by the binding of the inhibitor to the protein, as can be seen from figure 6a. The protein did reach some rudimentary form of stabilization post 13 nanoseconds. However, this was not as stable as the apoprotein, which can be seen there in black. The RMSF over here, represented by figure 6b, indicated large fluctuations in the positions of each of the residues of the protein, with an average change from 5.93 angstroms before binding to 12.07 angstroms after binding of the inhibitor, providing an indication that the protein no longer conformed to its native 3D structure, and thus would not be able to perform its function optimally. The SASA average prior to and after binding of the inhibitor was then calculated to be 7,686 angstroms and 7,662 angstroms, respectively. This decrease indicates that there is less surface area for the solvent to access and interact with in comparison to the apoprotein. And this is also good, as this also indicates reduced functionality of the protein. The ROG, represented by figure 7a, indicated a decrease in overall structure compactness, representative of the decrease between the average values calculated for the apoprotein and the bound protein complex, being 15.62 angstroms and 15.42 angstroms, respectively. This adds to the information provided by the RMSD and RMSF in terms of elucidating further the increased destabilization of the protein. And with regards to the hydrogen bonding, represented by figure 7b, the bonding formation capabilities of the protein alone and the protein in complex with the inhibitor is very similar, indicating little change in the number of hydrogen bonds formed. Okay, then figure 8 over here just identifies the molecular structure of dinorextin once again, and its generated pharmacophore structure. Following the generation of the pharmacophore and uploading this to the Zinc Pharma server, 11 pharmacophore hits were obtained, and the results thereof can be seen in table 2, where all 11 hits with their respective compound names have been displayed. So this is just 4 of the structures, and then here are the rest. Now the red, blue, green, and yellow highlighted elements in each of these proposed inhibitors or proposed hits correspond to oxygen, nitrogen, fluorine, and sulfur, respectively. Concluding findings of this project indicated that dinorextin portrays very good inhibitory characteristics, as well as a very strong binding affinity for the active site of the modeled axonemyl-dynein protein, and is a good target from which to have designed a pharmacophore. And what all of this means is that if a viable, strong inhibitor of the axonemyl protein target is designed using these and future results as the basis for it, the inhibitor, once made into a drug safe for human administration, could drastically reduce the number of new schistosomiasis cases occurring each day, as well as reduce the number of individuals at risk of contracting the disease. Now, to further this study, the top 3 identified hits will be determined through binding each of the identified inhibitors to the protein and performing molecular docking MD simulations, as well as Lipinski's rule of 5 on each of these systems to identify those with the most negative Z score, which then reflects the best affinity of the ligand for a specific binding site on the protein. And furthermore, the druggable compounds which provide the most promising inhibition to the identified protein will then be synthesized, followed by in vitro investigations. Okay, this is just the references, and I would like to acknowledge and thank Prof. Abidemi Paul Kapo, Dr. Priscilla Masamba, and Dr. Geraldine Munsami for their continuous support, guidance, and supervision of the research. Thank you.
Thank you. Thank you. It was a very nice talk. Are there any questions? Dauda, please.
Yes, good talk. So I have a technical question. So how long does it take you to run a simulation, and what kind of specs do you have on your on the machines you use?
Sorry, in terms of the second part of your question, I'm not too certain what you mean by specs.
How many CPUs or GPUs are you using on your computers?
Okay. So basically, the simulation can be run for longer. They can be run for 100 nanoseconds or 200 nanoseconds. But the reason we stopped at 60 was because the complex, once the inhibitor had bound to the complex, it reached some form of stabilization post 30 nanoseconds. So there's no point in continuing it past that point if it's reaching stabilization. And in terms of the specs, we uploaded the information to the CHPC server in order to run the simulations.
Okay. Yeah. Now, so I think my question was not clear enough. So, for example, so for your 60-second simulation, so how long does the computer takes to do that calculation, or the high performance server?
Okay, that
is it a few minutes, a few hours, a few days, or how long?
A few months.
Oh, okay. Yeah. Excellent. Yeah. So that's
there we go.
I wanted to know. Okay. Yeah. Excellent. Thank you.
Thank you.
Any other questions for the speaker? Well, let's give them another round of applause. So we'll just proceed with the next speaker on the program, but I do see that the very first speaker that was to be has joined the session here, and we'll just make him the last speaker. So our next speaker is down the list here. Fatiha. Yes, thank you. You were here earlier. Ah, here you are. Yes. How do I say your last name? Is that Fatiha Smain?
Yes, yes.
Thank
yes,
you.
Hello. Yes, I am Fatiha.
Yes, so you can share your screen and you can begin your talk.
Okay. Can I start now?
Yes, please.
Okay, so hello all. I am Fatiha Smain from Theoretical Physics Laboratory, Department of Physics, University of Abu Bakr Belqayed, Tlemcen, Algeria. The topic that I will present today is Poisson-Boltzmann equation for charged pellets. I am interested by the electrostatic interactions in soft and biological matter. In my presentation, I will cover the following points. First, I present briefly the Poisson-Boltzmann equation, then give its form for planar geometries, which has several applications in biological matter like planar membranes, and explain the diffusive double layer and ionic profile close to charge interfaces, and its relation with the screening phenomena, which is present in charge systems, and I give some results. So, matter is made up of charged objects. For example, in biology, we have DNA, proteins, biopolymers, biomembranes. In soft matter, we have like examples, colloids, micelles, emulsions, and in interstellar matter, we can find dusty plasma, which form 99% of the universe. The effects of the electrostatic interactions are important in the study of the behavior of matter. Understanding the electrostatic interactions acting in biological matter is very important because of its crucial biophysical applications. For example, the determination of forces between membranes, the calculation of the electrostatic potential near charged surfaces, etc. In biological systems are in most cases complicated for analytical treatment to provide solutions. In those cases, numerical procedures can be involved. In general, Poisson-Boltzmann equation is an approach used to quantify the electrostatic contribution in a system formed of charged objects immersed in a continuum medium. To introduce the Poisson-Boltzmann equation, or the origin of the Poisson-Boltzmann equation, is the Gauss law. This physical theorem says that if we have a number of discrete charged inside a surface, the total of the electric flow out of the closed surface is equal to the charge enclosed divided by the permittivity. This theorem is summarized by this equation. Because of the electric field A is a differential of the electrostatic potential, we can write this equation. And finally, we can give what we call the Poisson equation, which relates the potential or electrostatic potential to the density of charged particles. So if we can solve this equation, the Poisson equation, we have the potential from which we can derive most electrical properties of the system, like electric field, electric forces, energy and free energy. But the problem here is that this equation is difficult to solve for system of interest like biological systems. The second equation, the Boltzmann equation, can be derived as follows. In a system, for example, like water, which interests biologists, the charge density is the sum of the density of the solute, the fixed particles, charged particles like proteins, and the density of free ions like cell ions. The ion distribution themselves in the solvent according to electrostatic potential can be given in equilibrium state by a no relation, which is the Boltzmann equation. This equation relates the potential, the electric potential, to the density, the density of ions. For example, for salt ion, NaCl, we have the number of Q here is the valency. The valency for the NaCl is equal plus and minus N, and Ni0 is the density of the bulk. And the rho solute can represent the surface of a membrane, of surface of potential. And here we draw a surface of protein, for example, surrounded by the salt ions. And here we have draw the asymptotic behavior of the density of ionic salts. Plasmon-Boltzmann equation can be solved in some cases analytically. This is possible when the electrostatic energy is less than the thermal energy. But for systems of interest, this is not possible. That is why we prefer solve the Plasmon-Boltzmann equation numerically. The system that we have to solve is this system. But depending on the, yes? Okay, so I continue. Now, depending on the geometry of the the particle that we treat, we can we must write the mathematical operator nabla following the geometry imposed by the problem. If we have a planar distribution, the nabla take this form. If we have here, for example, this can be modeled the membranes problem. If we treat, for example, DNA, we can adopt the cylindrical geometry, and in this case, the mathematical operator can take this form. And if we treat globular protein, the nabla take this form. But unfortunately, we have to solve non-linear differential equation. Plasmon-Boltzmann equation need what we call boundaries condition. And generally, we have three types of boundary condition. We can consider the first class of boundary condition, first fixed charge surface. This can be expressed by the fact that the derivative of the electrostatic potential is equal to the charge multiplied by what we call Jerome-Land divided by the elementary charge. And and here I have utilized for technical implementation the reduced potential psi, which is equal the electrostatic energy divided by the thermal energy. When we treat one membrane model, the boundary condition adequate for the description of the system are those equations. This equation means that the derivative of the electrostatic potential, which is proportional to the electric field, is equal to is proportional to the charge the density of the membrane. And the second equation says that far from the membrane, the potential, there is no electrostatic influence. So we can write this equation. In other problem, or in the other situation, when we study the interaction between two membranes, we solve the Plasmon-Boltzmann equation on its linear form, and we take like boundary conditions those two conditions. Those two conditions significant that the potential, the surface of the first the charge of the first membrane is fixed by its electrostatic potential. Yes, electrostatic potential at the points XA, and the second equation fixes us the electrostatic potential, or fix us the value of the surface charge via its derivative of its electrostatic potential. Or when we treat the problem of the interaction of DNA and protein, we need some idealization. For example, if we are interested by studying globular protein. A globular protein can be idealized or sent as a sphere of radius r and surface charge density sigma protein. In a solution or in aqueous solution, this protein is surrounded by ionic clouds. And this defines what we call its double layer, electrostatic double layer. For DNA, we can idealize the DNA as a cylinder of radius r. And if we are interested by the interaction protein DNA, we can utilize this simplification. When we are interested by studying this system for the electrostatic interaction using Poisson-Boltzmann equation, so when the distance between the protein and the DNA is less than the characteristics radius of the protein and the DNA is when L is less than the radius of the DNA and the radius of the protein, we can consider the two surfaces as planar surfaces. So we can consider this complex system as the interaction of this complex system as the interaction of two planar systems, and there we can explore the equation of Poisson-Boltzmann in its planar form. Here I recall that this length is have an important role in when studying the electrostatic interaction. It gave us an information about the screening length, the effect of the screening length created by the surrounded ion or the clouds of ions around the charged pure particle. Here we give some biological parameters. For example, for planar membrane, the surface charge is equal 0.050 electron per angstrom square. For DNA, we have this value, and the charge of the DNA is negative, minus 0.01 electron per angstrom. And for protein like mica, I found this value. And here, after solving numerically the equation of Poisson-Boltzmann for this system, I can draw here the behavior of the potential around two surfaces. This X represents the first planar charge, and here the second planar charge, and here we see the behavior of the potential, the reduced electrostatic potential. And here I have drawn the potential for different value of R. R is a parameter introduced, R here is a parameter introduced to vary the value of the second density of the second plan, or the second membrane. For example, we see for R equal, if here the sigma of the first plan is equal to 0.01, and if R is equal to 1, the green line, continue line, we see here a symmetry. Here we have a symmetry, and varying R, when we vary R, we can vary the value and the sign of the density of the second membrane. Here, the corresponding electrostatic field for the same values, and here I have drawn the density profile of positive and negative ions. And I recall that I use the data of a monovalent salt, like NaCl. And here I have ejected in my program the value of the DNA. Here is the surface charge of the I draw the potential of between two surfaces. The first surface represents the DNA, and the second surface is a charged surface for which I have varied the value of the density of the second valve by varying R. And here we see that the potential vary, the variation of the potential depend on the value of the density of the second planar charge. Here I have drawn the electrostatic potential for the same data, and here we see the distribution of the positive and the negative ions cell between the two membranes. Because of the negative thing of the first membrane, we see that we have a small presence of a negative ion for all the values of R, and all the positive ions are closed to the surface of the potential. So knowing the value of the potential, the electrostatic potential, this permits us to calculate important quantities which can interest biologists, physics, and chemists, chemical. So thank you.
Thank you. Thank you for this wonderful talk.
Thank you.
Are there any questions for the speaker?
I have a question.
Please proceed. Go ahead.
Yeah. Okay. Thank you for a nice presentation. So my question is, it seems like you make very large approximations on the molecular shape, like where you talked about if you have a molecular shape that is spherical and you have a molecular shape that is cylindrical, you can approximate both shapes as planar surfaces interacting together. So the question I have for you is, do you have like an experimental data that you use to validate your theoretical model? Because if you make such big approximations, I'm just curious to know, like, how does that agree with experimental data?
Yes, I have chosen parameters. Plasmon-Boltzmann equation is largely utilized in physics and in biology. So here I have used parameters that have been validated in other works which exist in the literature. So my object today is to show how can Plasmon-Boltzmann equation modelize or treat biological systems, because Plasmon-Boltzmann equation exists, and there is data which explain that the validity, the Plasmon-Boltzmann equation gives a good results.
Any other questions? I have sort of one question going back to one of the previous speakers who talked about the electrostatic nature of the binding pocket for her enzyme. Now, there are some Plasmon-Boltzmann solvers that can solve, that treats each atom as a point charge, I think. Does the program that you're using and the solver that you're using, does it have that capability?
No, I have wrote my own program.
do you have the capability to make the domain a protein? So like if you have the coordinates of each atom, could you solve for that type of irregular domain?
So please, I am physics. I'm here for. I am with you, and I'm searching the parameters, biological parameters, which permits me to apply Poisson-Boltzmann equation. I have applied the Poisson-Boltzmann equation for example, when I'm physics. First, I'm physics. When I was studying density plasma, I have data and I have parameters, experimental parameters I can compare. But to compare data with the biological system, I start in this domain and I'm searching. I participate to this colloquium to have some collaborations and to apply my knowledge on the Poisson-Boltzmann equation.
Great. Well, thank you. Thank you.
Thank you for you.
Any other questions? So let's give this speaker another applause and a thumbs up. And so is there no other questions? We'll move on to Robbie, who was the first speaker, but he's here now and he's ready to give his talk now.
Yeah, thank you very much. I just need
so I think Fatiha, you need to stop sharing your screen, and
yes.
So Robbie, you can share your screen.
Okay. So thank you.
Give me a sec. Okay. So good morning from here. I am Rabi Darwish, a postdoc researcher at Harvard Medical School in the Department of Biological Chemistry and Molecular Pharmacology in the group of Professor Kevin Struhl. Today, I'll speak about a method I developed in the lab called PhenoRNA, which is a method that is used to link a specific phenotype to the genotype. The title of my talk is PhenoRNA, a method to associate a gene with a specific factor linked to cellular transformation. And specifically today, I will show you how from phenotype and by the use of computational biology and bioinformatics, we can uncover interesting genes that are cancer-related genes that they were never identified before. So this is the main goal of my presentation. While we might not be able to live without it, too much inflammation can cause serious damage. In 1863, the German pathologist Rudolf Virchow was the first to ask whether inflammation might contribute to cancer because he was the first to see some tumor samples in immune samples. After that, different clinical and epidemiological studies start to show a strong connection between inflammation and cancer. For example, in breast and cancer cells, they showed that the interleukin-6 was highly upregulated in these cells. Another example, the oncogenes RAS and MYC were also activated the master transcription factor and FKB via interleukin-1 beta. Another example, it was the SERK, the oncoprotein SERK kinase, activate the signal transducer and activator transcription 3 via interleukin-6. And this one, SERK, it's frequently activated in malignant cells, and it plays a role in regulating many genes crucial for cancer inflammation in the tumor microenvironment. So different studies, they told us that it's correct. There are many links and association between inflammation and cancer, but the direct mechanism in how inflammation can induce carcinogenesis is still unknown. Previously, our group of Professor Kevin Struhl described an experimental model of oncogenesis, where they use breast cells, MCF10, called MCF10A, but these cells were coupled to the ER circ, which is the fusion between the androgen receptor ER coupled with the oncoprotein circ. At this stage, as we can see here, the MCF10A cells are not transformed, so they are healthy and normal. But after we add tamoxifen to the media, to the cells will become transformed after 36 hours, as we can see here. So the tamoxifen will activate the oncoprotein circ, and the cells will change their status from non-transformed to transformed cells. What's important is after 36 hours, so if we will remove the tamoxifen, the cells will stay transformed. So the phenotype is maintained, even with the lack of the tamoxifen. So it's with the lack of the original cause of transformation. So from this experiment, we can conclude that the circ activated of the circ oncoprotein converts non-transformed breast epithelial cells into a transformed cells station. They also showed that the maintain of the cells from non-transformed to transformed cells involved many key factors, in particular NF-kappa B, LNA28B, LED7 microRNA, and interleukin-6, and all of them plays an important role to link inflammation to cellular transformation. So as we can see here in this figure, once the circ is activated, it will activate the transcription factor NF-kappa B. So once it's activated, it will also activate the interleukin-6 via direct transcription or indirectly via activation of the LNA28B that will inhibit LED7. And in this case, interleukin-6 will be highly regulated, and its own upregulated will itself also activate the NF-kappa B, and this will maintain a positive feedback loop that maintains the transformation state. Our lab also showed that in different studies in our lab that this positive feedback loop is far more extensive, and our lab worked intensively to generate a comprehensive elucidation of transcription regulatory circuits during transformation. And as we can see here, they showed also that there are many genes, exactly, it's a 27 gene that they are important in maintaining this, this transformation state, and they were responsible in linking this inflammation to transformation. My goal was we wanted to try to find another method or another way to provoke transformation, but without using the circ protein. So I was looking to find a new method of provoking transformation in the big goal to identify and validate important genes and pathway that important in the model of cancer system. And for that, I needed to find a new way. And the idea was I wanted to induce transformation by adding extrinsic molecule to the cells and to try to mimic the tumor microenvironment in more accurate way. So to do that, I needed to find a way to measure the transformation, and I was using GILA, which is the grow in low attachment, which is able to give me a quantitative metric of transformation. The GILA, it's very simple. It's very similar to the soft assay, which is used for 50 years old to study transformation, but this one is most faster, and it's feasible to do higher big amount of screening. I will show you the principle of it. So in the day one, I will treat my cells, which are the MCF10A parental cells. So I will not use the MCF10A, which is fused to the ER circ. It will be only the parental cell MCF10A. So in these cells, I will add extrinsic molecules. I will show you in a bit. and then I will see these cells after 5 days if they are able to grow in low attachment cells or in the high attachment cells. So if the cells are transformed, so they have the possibility they can grow in the low attachment cells and they grow in suspension. If the cells are not transformed, they cannot grow in low attachment plates. And at the same time, I will be using the high attachment cells as control. And in the high attachment, most of the cells, transformed or not, are able to grow. After growing, I will measure the ATP, which give an idea if the cell growth or not, and I will make the comparison between cells are able to transform or not. So briefly, if we will see to this one, in low attachment, if the cells are transformed, so they grow. If they are not transformed, they don't grow under low attachment condition. So now what I will do is I will treat the parental cell MCF10A with different molecules, such as light, LPS, oncostatin M. I will treat the MCF10A parental cell even with individual cytokine or even in combination between 2, 3, 4, or 5 conditions. So I screened around 100 of conditions to obtain cells transformation. And as we can see here, for example, here it's the cells that they were able to transform and other cells that they were not able to transform. Out of the hundreds of conditions, I select 17 conditions of transformation that they go from weak or mild transformation to moderate transformation to very strong transformation. And from that, I create what I call phenotypic series. So as we can see here, the first diagram, it's under low attachment. And here it's the MCF10A that they are growing with different conditions. As we can see here, the MCF10A cells, so the parental cells, they are not transformed. They are not able to grow, which is very logic because it's under low attachment. Only transformed cell can grow. And for the 17 conditions that I picked, we can see, for example, for LPS alone, it has no effect in the MCF10A cells and they are not transformed. While certain mix of transformation using different cytokines that they induce different signaling pathway in a matter or another, we can see that transformation starts to be stronger with different conditions than another. For example, the Q condition, which is the mix between oncostatin M, EGF, and tumor growth factor alpha, they showed very strong transformation, as we can see here. So from here, we created a phenotypic series of transformation going from weak to strong. And if I will show you the comparison, which means the growth in high attachment, as we can see, all the cells are able to grow because transformed and non-transformed cells are able to grow here. So from this experiment, we succeeded. This treatment showed us a wide range of transformation phenotype, but they do not affect the growth in standard conditions. So we are very confident with these conditions. After that, what the idea was, we needed to link this phenotype to the genotypic data. So what I did, under all this condition transformation, I performed RNA sequence, and like this, we will obtain as much as possible information about the transcriptional profile. How we do it? So under every conditions, I purify, under this, every cells treated with its particular conditions, I purify the RNA, isolate them, then I prepare the library, which means convert the RNA to cDNA, and we add sequence adapters. Then we check the library quality using the DNA chip on bioanalyzer. And at the end, we send it for sequencing using Illumini sequencing. What we got back from the information that we obtained that, which was expected results under every conditions. We have some genes that they were upregulated and some other genes that they were downregulated. As we can see here, for example, in condition A, 1,000 of genes were upregulated comparing to the non-treated cells, while other cells were downregulated also comparing to the non-treated cells. Conclusion from here, the RNA-seq experiment, they showed us different genes expression under these experimental conditions. So now we have from one side all this phenotypic data, and from another side we have the genotypic data. And I wanted to take as much as possible information from these two data to uncover new genes that they are important in cancer. It's correct that there are variety of whole genome approach that can determine which genes are important for specific phenotypes. And also, alternatively, there are systematic genome scale screens that can also help to identify genes that are affect a given phenotype. But these mutations may cause such phenotypes by artifact or by indirect effects. So my method that I'm telling you today, I called it's pheno-RNA. And this method, it's only correlate the transcriptional profile, as we can see here, to the simple gene, to the strain of the phenotype. So every gene is linked to the power of transformation. And from here, I obtained what I called pheno-RNA. Pheno-RNA, it combines the high throughput mRNA-seq with a precise readout for the phenotype. And this one I will be using to identify the candidate genes that they appear to be critical for oncogenic effect of the transformation. So the pheno-RNA, first, why it's a novel and it's a new technique, because it set a quantitatively measured phenotype ranking from null to severe, and the experimental profiles of these individual genes are then correlated with a quantitative measurement of the phenotype. The expression profiles of genes driving the phenotype should be highly correlated with the strength of the phenotype. So upon the RNA-seq analysis for the almost 6,000 genes that I obtained, I determined the Pearson correlation coefficient between the mRNA levels and the degree of transformation under the 17 experimental conditions. As we can see here, the Pearson correlation, it explained to us how much a phenotype is strong or weak. For example, when we make this correlation between the phenotype and the genotype, if the correlation coefficient is between 0.8 and 1, it means the transformation to the transformation or the genes is very strongly important to the transformation. If the correlation coefficient is between 0.6 and 0.8, that means that the transformation is the link between the gene to the transformation is strongly correlated. While if it's below 0.6, so the gene is moderately or weak correlated to transformation, and below 0, it's negative, and also it needs to be minus 0.8 to minus 1 to show it's very negatively strong correlated to the transformation. As we can see here from this experiment, that with pheno-RNA, we found around 200 genes that they were identified, and their expression profile are remarkably correlated with the transformation phenotype. As we can see here, we have also many genes that they are strongly correlated to transformation, but the phenotype with the negative gene was less dramatic. So from this experiment, we find that there are 200 genes that they are strongly correlated to transformation. The first question that I asked after is to see if these genes are really matter for transformation, are really important. What was easy is to run, I was using a different websites and databases like David and BioPortal to help me to see which pathway these genes are involved. And as we can see in this diagram, it's correct that these 200 genes are strongly associated with already known cancer pathways. For example, if we will look in this inflammation A, which is the red graph, we can see that the genes that they are highly correlated with transformation are very enriched in the inflammation pathway, which is correlated to cancer. Another example, if you will see the cell adhesion, also the genes that they are highly correlated with transformation are important for cancer, and they already known to be involved in cancer pathway. From this experiment, we can say that it's correct that these 200 genes, like what it's already known from these 200 genes, are very associated with cancer. As we can see here also, the genes that they are correlated with transformation, they are also upregulated in cancer-associated cells, while the genes that they are not correlated with transformation, they are not associated with cancer pathways. Conclusion, genes whose expression profiles are highly correlated with transformation are correct, and they are enriched in cancer-associated pathways. And also, what it was important from this technique, that it's a discriminative method. For example, it didn't just tell us which genes are upregulated or downregulated. For example, we have the data from previously from RNA-seq of the MCF10A ER-circ that also they show transformation phenotype, but the microRNA and the RNA-seq, they only told us what genes are upregulated or downregulated. But this technique, when I did this Venn diagram here, we can see that, for example, here I have the 200 genes that they are upregulated to transformation, but they can literally explain to us from the other method which genes really matter for transformation. For example, we have almost 550 genes that they are upregulated in the microarray and the RNA-seq from the ER-circ, but 39 only matter for transformation. I will give you also one example of this. For example, I picked here only the inflammatory genes. For example, as we can see in the blue colors, these genes are upregulated in both methods, in mypheno-RNA and in other method. So they showed what it was previously discovered, and they confirmed what it was also discovered before. They also showed that other genes that they were upregulated in another method, but they were not shown by ours. But we can tell now that if they are important or not for transformation. And here in red also we can see many genes that they are strongly correlated with transformation. They were found by other method, but it didn't show up in others. Conclusion, this pheno-RNA, it helped us to distinguish among genes that are not strongly correlated with the ones that they are or not strongly correlated with the transformation phenotype. So now, after we know that these genes are fully enriched in cancer genes, the next question was if we can find a subset of genes that are most relevant to this transformation and they might be important for cancer. For this one, I used bioinformatics and different available databases to answer to this question. So what I found till now, as I told you, I have around 200 genes that they are very strongly important for transformation. What we did, it's a screening for non-cancer related genes. First, we can use PubMed, which is like by using NCBI utilities, and it will give us if a gene was already studied linked to cancer or not. And after that, after obtaining the list, I did a manual and the literature review to confirm if a gene is studied before or not in cancer. After that, I check cross my data with available databases such as the network of cancer genes, cancer gene census, and tumor-associated genes to see which genes are important or not for cancer. And from this, I obtained around 90 genes that they are very strongly important to transformation, but they were never studied before in the cancer field. And these 90 candidate genes are of a high importance, and it will be studied furthermore. So the pheno-RNA identified the genes that they are highly correlated to the transformation phenotype, but they have never been previously linked to cancer. we found that some of these genes are important in many pathways, such as immune response LTBR1 signaling. For example, this pathway, it's known that it's important for cancer, but we found new genes in this pathway that they were never linked before to cancer. Also in chemotaxis CCR1 or in signaling transducin adenosine 1 receptor signaling pathway and others. And as I was speaking inflammation, I want to show you this diagram just to see pheno-RNA, how it's helping us. We know that we have obtained around 200 inflammatory genes in our screen. For example, 19 of them, they were highly important for transformation. 13 of them, they were already studied, and 6 of them, they were not studied before, and they are highly potential to be very important genes in cancer. 4, the 6 of them, they came up with our technique, pheno-RNA, and 2, they came from other studies. So this pheno-RNA, they uncover also different genes that they were never studied before. And this suggests that they are potentially unknown genes that play a role in cancer. So here it was how I used computational biology and bioinformatics to link phenotype to transformation. And at the end, we obtained a set of imported genes that they might be important for cancer. To conclude, pheno-RNA, I hope I convinced you that pheno-RNA is a general approach to identify candidate genes associated with a specific phenotype by generating a phenotypic series by treating non-transformed breast cell line with a wide variety of molecules that induce cellular transformation to various extents. We identified by using this genes whose expression profiles are remarkably correlated with transformation phenotype, and they are enriched in cancer-associated pathways. This novel approach of transformation paved the way for the identification of a new cancer candidate genes that have never been linked to cancer. And also, pheno-RNA can be used also for many other phenotypes. For example, they can used by generated to study, for example, morphology or focus formation, cell motility, invasive growth, and metformin sensitivity. And also, for example, I use this molecules or the cytokine that I need to provoke transformation, but also everyone can use it to add whatever they need related to their experiments to make this set of data. In the future, what I am doing now is to validate. I selected different genes that I am working on them, especially 2 that they are shown that they are very important transcription factor. I would like to validate the oncogenically relevant target genes of them by using siRNA and CRISPR. I will molecularly dissect the regulatory circuitry that these targeted genes affect as it relates to transformation. And also, I will link my data to previously existing regulatory mechanism and circuits. And it will be also interesting to analyze our cancer candidate genes in cancerous and normal tissues. At the end, I would like to thank you for your attention. I would like to thank Professor Kevin Schrull that I'm doing my postdoc in his lab. I would like to thank my lab members, in particular, Joseph Geisberg and Zarmit Moqtadiri, our lab manager, Catherine Maddox, my collaborator in Dana-Farber, Asaf Rotem, and the Swiss National Science Foundation and the NIH for their funds. And thank you very much for your attention.
Wow, what a really interesting talk. I'm glad you were able to make it to this session.
Thank you very much. I'm sorry for not being...
Oh, no problem. It was very much worth the wait.
Thank you.
Is there any questions? Are there any questions for this speaker? I have one question.
Yes.
Could you extend the method to epigenetic features? Like sequences in the histones and or the histones.
Yes, it depends also, for example, when you will read the sequence, it's mostly, I think it's mostly when you would like to read. So for me, I was not looking for these histones, but I think, yeah, it's feasible by using a correct software for this.
Wow, it's a very powerful technique. Very powerful.
Yes.
Any other questions for the speaker? So you are currently at Harvard?
Yes.
Okay. In a permanent position?
Yes, I am. It's my 3rd year of postdoc now.
Got you. Okay. Thank you.
Thank you.
Well, if there's no other questions, this brings us to the end of this session. And in a few moments after the end of the session, we'll be sending out a conference survey. And I'd ask that people answer those questions as quickly as possible, because we have an open forum tomorrow on the future of biophysics in Africa. And we'd like to discuss the initial results from the survey. So you'll be getting that by email in a few moments. And if you could address those questions as soon as you can, we'd very much appreciate it. So if there's no other comments or questions, let's thank all the speakers again.
Thank you.
I'm going to do the clap here. Yep. And in addition to the clap, I'll do a thumbs up and love. We need more love in science. All right. Thank you, everyone. And we'll see you tomorrow morning.
Thank you.
Or I guess we'll see you. I guess there's no morning session tomorrow. It's tomorrow afternoon. Or tomorrow at noon is when the open forum is. That's noontime GMT +2.
Okay.
All right. Thank you, everyone.
Thank you.